Hello everyone, welcome to episode 53. Today I'm continuing my series on plant physiology by exploring the subject of reproduction, how plants reproduce and perpetuate themselves. Although there are numerous kinds of plants, from green algae to mosses and liverworts to huge redwood and spruce trees, there are also angiosperms, or flowering plants. I'm going to mostly focus on the reproductive physiology of flowering plants, because angiosperms are the most common kind of plant on the planet. They're the most useful, and the most harvested kinds of plants, and they're just about the most interesting and dynamic kind of plant when it comes to reproduction, because they have these really unique, beautiful flower structures that have so much variety from species to species. When I was talking about plants in episode 33, I mentioned the complex alternation of generations part of plant reproduction. It's an integral part of their reproductive cycle. And because it's so integral, I'll explore that concept again in the beginning of this episode, pretty much right now, because it's fundamental. This alternation of generations means that as a plant lineage reproduces and perpetuates itself, each subsequent generation kind of alternates back and forth between one of two cellular phases or forms. In Series 3, I talked about genetics. I discussed that we humans are diploid organisms because our cells have two copies of our genome. We have 23 different chromosomes, and because we're diploid, the prefix di, meaning two, and ploidy, in reference to the number of chromosome copies, we have two copies of our 23 chromosomes for a total of 46 chromosomes in each of our cells. Now, in plants, one of the two cell types in this alternation of generations is also a diploid organism. It's called a Diploid Sporophyte, or the Sporophyte stage, and it has two copies of its chromosomes. Now, the other cell type is a Haploid Gametophyte, and this means that this generation, this individual, only has a single copy of each of its chromosomes in each of its cells. The sporophyte and the gametophyte are so named because of the reproductive cells that they produce. The diploid sporophyte produces spores, and the haploid gametophyte produces gametes. So within the sporophyte are structures called sporangia, and these are reproductive structures, and the cells within them, the diploid cells within them, are undergoing meiosis, and this produces haploid spores, or spores that just have a single copy of the plant's DNA. These spores are released into the air, where they land somewhere, and if the ground is fertile, if the conditions are right, they will germinate and grow through mitosis. Without any kind of fertilization or input from another plant, this spore will asexually grow into a new haploid gametophyte. All of the cells in the gametophyte are haploid, and so all it takes is simple mitosis to produce more gametes, because they're just haploid like they would be in a diploid sexually reproducing species. The nuance here is that some of these gametes are sperm analogs, while other gametes are egg analogs, and these create kind of a rough male-female differentiation in the gamete generation. These haploid gametophytes, whether male or female, will produce sperm or egg gametes, respectively. When a sperm and an egg gamete meet and fertilize, their haploid chromosomes will fuse together and create a diploid sporophyte zygote. The cells in this zygote divide by mitosis, growing into the sporophyte's mature body, which eventually grows specialized reproductive cells called sporangia, and these sporangia produce and release more spores, and the alternation of generations begins again. The sporophyte and the gametophyte can be wildly different in size and shape when you compare different plants. There's a huge variety to the body plans, to the life strategies. For example, in angiosperms, the sporophyte is the main phase of the plant's life. It's the main body structure that grows the largest and lives the longest. If you're looking at a flowering plant with flowers blooming and open, you're looking at the sporophyte stage of that species. It's a diploid organism. The sporangia in the sporophyte is composed of two structures, called an ovule and an anther. They both produce spores, but each produces a specific kind of spore. The anther produces microspores that will become male gametophytes, while the ovule retains megaspores that become female gametophytes. When the microspores are released from the flower, they become pollen, and the pollen is meant to land on some other plant's flower so it can pollinate the megaspore that's being kept in the ovule there. This fertilization of the microscopic haploid gametophytes produces another diploid sporophyte. In mosses, this priority is reversed. The gametophytes are large and long-lived, and the sporophyte is tiny and dependent on the gametophyte. In moss, the sporophyte is a thin stalk, growing out of the female gametophyte with a little sporangia on the end. The sporangia releases male and female spores, which grow into male and female gametophytes. These are the main structures of the moss, the main fuzzy tendril of the plant. At their very tip, the male gametophytes grow sexual structures called antheridia, which release a cloud of tiny sperm into the air. These land in the analogous structures at the tip of the female gametophytes, which are called archegonia and contain the eggs. The sperm enters the archegonia and fertilizes the egg, and this creates a diploid zygote. This zygote then grows into a thin sporophyte stalk, and this grows out of the female gametophyte and it produces more spores, thus starting the cycle all over again. Flowering plants provide the most interesting angle on plant reproductive structures. The flower is bright, it's fragrant, it's attractive to bees and humans alike. I talked about flowers briefly in episode 49, mostly about the gene expression involved in flower development. So today, I'm going to cover some of the same basic details, but I'll talk more about the physiology and the function of the flower as opposed to its genetic expression. There are 4 distinct parts to the flower. There are the sepals on the outside, the flower petals themselves, the stamen structures that release spores, and the carpel structures that hold the ovary. As you presumably know, flowers have a huge diversity in appearance. Not every flower has every one of these parts, and the size and the shape of the sepals, the flower petals, all of it, there's a lot of variety between species. The sepals are the outermost layer of the flower, and compared to the petals, the sepals are much more leaf-like. When you're talking about all of the sepals in a particular flower, you're talking about a structure that's called the calyx, the flower calyx. The purpose of the sepals, or the calyx, is largely protective. They're thicker and sturdier than the petal tissue, and when the flower is growing and getting ready to bloom, the sepals are wrapped around it. They're wrapped around the flower petals and the reproductive structures within it, and they provide a degree of mechanical protection from bacterial and fungal pathogens, as well as herbivores like insects who would want to eat at the soft flower petal tissue. In many cases, even after the flower is bloomed, the sepals continue to provide some measure of protective or structural support. The flower petals are the next layer inside of the calyx. As you almost certainly know, flower petals are the flashiest part of the flower. They have all the beautiful color, and they exude the scents that make the flower smell so good. There's a whole industry about selling flowers so people can enjoy their fragrances. That's how good it is. The reason flower petals have these attractive qualities is because they need them to support a critical role that flowers play in plant reproduction. Angiosperms have evolved flowers as part of a symbiotic relationship to attract various flying species of pollinators, like bees or hummingbirds. The plants found that getting a little animal to carry your pollen from flower to flower was actually a really effective way to reproduce, far more effective than just throwing your pollen out into the wind and hoping for the best. The colors of the flower petals are used to visually excite an animal, to get it to notice the plant and start paying attention to it and maybe zone in on it because they're attracted to it. And the scents are used to lure them in, to guide them to the center of the flower and lead them to where the pollen is. So flowers use a lot of these attractive qualities to kind of lure in pollinators because it helps them reproduce. Some plants use nectar to incentivize pollinators, which is produced by little glands called nectaries that exist at the base of the flower petals. The nectar is a sugary fluid that animals like hummingbirds and butterflies depend on for food. These animals are called fluid feeders, and the fluid that they eat, the fluid that they feed on, is nectar. They drink the nectar through a long tube organ called a proboscis, which has actually created a kind of mini evolutionary arms race between plant and pollinator. The nectar is sugar-rich, and it takes a fair bit of sugar resources to produce it. It's expensive. As such, the plant benefits the most when this nectar can be used to entice multiple pollinators. If one pollinator comes by and drinks all of the nectar, then all of that nectar, all of those resources just went into one animal, who may or may not have even carried off any pollen to help the plant reproduce. In this case, the plant just wasted all of that expensive nectar. So instead of this, the plant wants multiple pollinators coming and each getting a little bit of nectar, and each one thus increasing the potential reproduction and spread of the plant. If the plant can get more pollinators to come by, it has a better chance at spreading its pollen and reproducing. So to prevent one greedy hummingbird or a couple greedy butterflies from taking all the nectar, some flowers have evolved a morphology where the flower petals make deep, narrow basins, with the nectar at the very bottom. The farther away the nectar is, the harder it is to reach, and the less nectar that any individual pollinator will be able to take. But because these fluid-feeding pollinators depend on the nectar for food, they then experience a very strong evolutionary pressure to grow longer proboscis, and this only encourages the plants to grow deeper flowers, and then that causes the animals in response to grow even longer proboscis. This is the essence of the evolutionary arms race, and it creates flowers that are comically deep, and it creates butterflies and hummingbirds with proboscis that are comically long, sometimes multiple times longer than their actual body is. Just like all of the sepals are called the calyx, all of the flower petals together are called the corolla. Inside the corolla of flower petals, there's the two innermost layers of the flower, and these make up the flower's reproductive structures, the actual reproductive cell-producing organs. The outermost of these two innermost layers is made up of the stamen, and the stamen are reproductive structures composed of a tall, thin stalk called a filament and an anther at the tip. The anther is a bulbous little organ that produces pollen, and when the wind hits it, or it gets rubbed apart by a pollinator like an animal or something, it'll open up and release pollen. The pollen can either catch the air and float into another plant's flower, which isn't particularly efficient, or it can stick to the hairs or the feathers of a pollinator and get carried that way as they move from flower to flower to flower. The innermost layer of the flower has reproductive structures called carpels, which are composed of a stigma, a style, and an ovary. For pollination to occur, pollen has to go into the carpel through the stigma, which is the upper portion, and then the pollen has to go down through the style, and the style is like a tube or a half pipe that just guides the pollen down into the ovary. The ovary contains little structures called ovules, which themselves contain the germ cells that undergo meiosis to create eggs. The pollen are sperm gametes, as they're also created through meiosis. Pollination is thus the process of plant fertilization, when this pollen sperm is kind of shuttled down the carpel to the egg in the ovary. Pollination is an interesting subject. Many species of angiosperm have flowers that possess both stamen and carpels, and each individual flower produces both pollen and eggs. Other angiosperms, called monoecious angiosperms, have both stamen and carpels, but the individual flowers that they have only express one or the other, not both. Both of these kinds of angiosperms are hermaphrodites, because they can self-pollinate themselves to create seeds. And while this is really easy for them because they have all the parts ready in one spot, ready to go, it comes with the penalty of lower genetic diversity. Then you have the Angiosperm plants, whose flowers only express one reproductive structure or the other across the whole plant. These are dioecious angiosperms, and because they have only one reproductive structure across the whole plant, they're basically male or female plants. The male plants have flowers that will produce stamen, and the female plants have flowers that will produce carpels. These dioecious angiosperms can't self-pollinate because an individual plant only possesses one reproductive structure or the other. They don't have both. They're not hermaphrodites. And so they have to rely on pollinators or the wind to carry their pollen from plant to plant. Even though this makes reproduction for them logistically more difficult, It offers a fitness benefit in that there's a healthy amount of allele mixing, which preserves genetic diversity. Plants with hermaphrodite flowers have evolved a few mechanisms to avoid excessive self-fertilization, and thus avoid the risks and problems associated with inbreeding. In some cases, the cellular maturation of the male and female gametes takes place at different times, so they can't self-pollinate because only one kind of gamete is ready at any given time. For example, imagine a plant where the pollen is mature and it's ready to be released, but the eggs aren't mature yet, and so they can't be fertilized in the first place, and so even if the plant does release its pollen, it simply can't fertilize itself. Some plants use molecular mechanisms, like some kind of signaling or identifying proteins on the gamete cell membranes. When the pollen and egg from the same individual touch, the membrane proteins on one gamete identify the other gamete as coming from the same individual, and fertilization is rejected. Other plants just use a brute force method. They simply position their anthers and their stigma such that they're really far apart, and accidental or unintentional self-fertilization is pretty much impossible. It's simple, it's effective, and it doesn't require any fancy membrane proteins or the meticulous timing of cell maturation. Okay, so how does this whole fertilization process work on a chemical level? In most plants, fertilization is simple, like it is in animals. Sperm meets egg, bam, done. But in angiosperms, this process can be a little more complicated. It involves not one, but two separate fertilization events. Before I explain that, I should really quickly talk about how sperm and eggs are formed, because how they form is relevant to how they reproduce. Inside of the anther, the structures that make pollen, there are diploid cells called microsporocytes, and these microsporocytes undergo meiosis to produce haploid microspores. Each microspore undergoes mitosis to split in two, creating a tube cell and a generative cell. Both tube cell and generative cell exist within a single pollen grain, encased in a protective, watertight outer coating. Now inside the ovule, there's a really big diploid cell called a megasporocyte, which divides by meiosis to produce 4 smaller megaspores. For some reason that's not particularly well understood, 3 of these 4 megaspores will spontaneously decay, leaving a single megaspore behind as the sole survivor. This surviving megaspore will expand to become an embryo sac, and the embryo sac will divide by mitosis to produce 8 haploid nuclei. 3 of these 8 nuclei will group up at the top of the embryo sac, 3 more will group up at the bottom, and the 2 remaining nuclei will just remain in the center, and these are called the polar nuclei. When a flower releases its pollen, the pollen grain gets carried by the wind or by some kind of pollinator animal to another flower, where it can land on the stigma of its carpel. The stigma is kind of sticky, which makes it easy for the pollen grain to be captured. Once it's been captured here and held here on the stigma, the pollen grain absorbs water, much like a seed does, and it germinates. Although a pollen grain is much smaller than a seed, so this germination process is similarly scaled down. Instead of a root coming out of the seed, the tube cell inside of the pollen grain will literally expand into a very long, thin, single-cellular tube, and it snakes its way down the stigma and down the style towards the ovary. The generative cell inside the spore is carried down the pollen tube, where it divides into 2 sperm cells, and these sperm cells flow down the pollen tube to the ovary. The sperm is deposited inside the ovary, where it makes contact with the egg and initiates fertilization to create a zygote. So this is the first fertilization event. But you'll remember that I said angiosperms do this fertilization thing in a really weird way. There's that second fertilization event, and that's because there's 2 sperm that go down the pollen tube. That generative cell divides into 2, and both of those little daughter cells are the sperm cells. The first sperm will fertilize one of the nuclei on the ends of the embryo sac, on the top or bottom. But the second sperm goes deeper into the embryo, and it fertilizes both of the polar nuclei at the center. This single sperm fertilizing 2 nuclei will create a triploid structure called the endosperm. This endosperm will become a nutrient-rich tissue that's used to feed the zygote in the seed during its germination. The endosperm is kind of like a placenta in mammals. It's a tissue full of proteins and lipids and starches that the developing zygote taps for nutrients and chemical energy, and it helps the zygote to grow at a time when it's obviously incapable of getting nutrients on its own. When a seed forms, the zygote and the endosperm grow to fill the ovule, and the ovule's outer cover hardens into a seed coat. While the seed forms inside the ovule, the ovary outside of the ovule begins to change as well. The ovary tissue accumulates sugar, and it swells, growing into a fruit. I talked about seed formation in episode 49 on plant development, but because it's a part of plant reproduction, I'll go over the process of seed embryogenesis again here. If you recently listened to that episode, some of this might sound familiar. So the zygote divides into two cells, an apical cell and a basal cell. The position of these two cells relative to each other determines the vertical orientation of the plant's growth. It establishes a developmental axis to the embryo, right away with the first cellular division. The basal cell will go on to divide into two cells. One becomes a cell in the root tip, and the other cell becomes the first in a chain of cells, like an umbilical cord for the plant embryo. The apical cell will go on to divide and produce the vast majority of cells in the plant. These apical cells will divide into three distinct cell lineages that create the three main tissues of the plant. The procambium, which becomes the plant's vascular tissue, the ground meristem, which becomes the plant's ground tissue, its pith and its cortex, and the protoderm, which develops into the epidermis layer of the mature plant. After a short period of cellular division and growth, distinct structures will start to emerge on the zygote. The lower part of the plant forms the radicle, which is the embryonic root. The middle part is the hypocotyl, which will form the stem. And then there's the upper part of the plant that forms the cotyledons, which become the first two leaves. You might be familiar with the terms monocot and eudicot. This refers to the number of cotyledons in the seed. Eudicots have two cotyledons, two of these embryonic leaves, and in most eudicot species, these generally absorb nutrients from the endosperm. Inside the seed, the cotyledons grow to wrap around the rest of the plant, creating a layer of soft green tissue between the endosperm and the hypocotyl. Because of their position as an interface between the seed coat and the embryo, the cotyledon tissue absorbs the nutrients out of the endosperm and becomes a new storage organ that can efficiently shuttle nutrients through the embryo to feed its growth. In this way, the cotyledons and eudicots can act as the primary nutrient storage organ instead of the endosperm. Monocots, on the other hand, have just one cotyledon, one embryonic leaf, and these simply tap the endosperm for nutrients instead of absorbing their nutrients entirely to take on the role of storage organ. When the embryo has fully matured and all the tissues have differentiated and developed into vasculature, ground tissue, and epidermis, the seed is ready to go. But it can't just exist in a wet stasis forever. It would run out of energy and die and rot. Instead, the seed dries out, and its cells stop growing. For all intents and purposes, the seed is now in a dry stasis, and it waits until conditions are right before it germinates. Recall how I said that when the seed develops in the embryo sac, the ovary outside the cell develops into a fruit? The fruit is a really useful thing for the plants, and from an ecological perspective, fruit is really useful for the local community. For plants, the fruit is a soft but protective shell encasing the seeds, and they serve as a mechanism for distributing the seeds outward in a greater geographic range. There are a lot of really creative ways that plants have adapted their fruits to serve these purposes. In many cases, the fruit just dries out, and it falls off the tree, and then it rots on the ground and exposes the seeds. That's the simplest method, and it comes with the seeds growing in a pile of decaying fruit, which is kind of like a little pile of nutrients, so that's nice, but it doesn't really spread the seeds out very far. It doesn't get the offspring very far from the parent plant, and so it's kind of limited utility, because you gotta spread out to thrive. Some plants have evolved really unique fruits that have really creative ways of solving this problem. Some fruits literally develop an internal pressure that causes them to explode, and the explosion will spray the seeds out and away from the parent plant. For example, the dwarf mistletoe fruit fills itself with sugar, and thus pulls in water through osmosis, which eventually results in a kind of controlled explosion because the water pressure inside just got too high, and this spews seeds everywhere. The sandbox tree is another example because it has fruit that will dry out as it ages. As the fruit gets older, the husk will dry out, and through this drying process, it'll shrink. Eventually, it'll shrink so much that the pressure of the seeds pushing against the husk from the inside will cause it to rupture, and the seeds can get thrown tens of meters away. A particularly popular and effective vector for seed dispersal is by using an animal. Animals are attracted to the sweet smell of fruit, and they'll gobble it up off the branch, or in the case of primates, they'll pluck the fruit off of the branch and eat it right out of their hands. The animal either haphazardly discards the seeds, or it consumes them along with the fruit and then poops them out somewhere else. In this case, the pooped out seeds come out in a thick pile of fertilizer, which promotes the success of the plant after germination. It's like starting with a silver spoon in your mouth, but for plants. While some animals eat the fruit and poop out the seeds, other animals, like mice, might just eat the fruit and the seeds. And when the seed gets chewed up, it's dead. It's not going to be able to germinate and grow into a new plant. It's destroyed. To discourage animals like mice and other rodents and stuff from eating their seeds, some plants have evolved defenses in their fruit structure, like the capsaicin in peppers that will burn an animal's mouth and scare it off. I should mention that there are several types of fruit. The most common type are simple fruits, like strawberries, coconuts, and radish, which develop from a single flower with a single carpel structure. Then there are aggregate fruits, like raspberry and blackberry, and these are multiple fruit structures that are formed from a single flower. Each individual flower has multiple carpel structures, and each of these carpel structures can produce fruit simultaneously. So you'll have one flower, but a lot of little smaller berries or smaller pieces of fruit. Then there are the weird ones, the multiple fruits, and these are like pineapple and figs. These are weird because the single fruit structure will form from multiple flowers. The flowers create initial growths that meet and then merge. They literally fuse on a cellular level to create a larger growth structure. This growth structure will turn into the mature fruit. Some fruits are intended to dry out as they mature, like a cashew or a walnut. Some fruits are relatively solid and dense, like apples, pears, and peaches. Other fruits are really wet and fleshy, like tomatoes or oranges. But in every case, the fruit tissue itself is derived from parent tissue, and it wraps around the seed, which is offspring tissue. The last part in plant reproduction involves the seed detecting when conditions are right, and then germinating. But how does the seed do this? How does the dry, inactive seed sense its environment? In warm, humid climates, especially tropical climates with no real seasonal differences and a perpetual growing season, seeds aren't adapted to be in any kind of dormancy. They can grow all year round, and so there's no reason for them to be dormant. Anywhere they land, it's going to be relatively fertile. It's going to be relatively moist, just because of the stable climate. But in more temperate and boreal climates that have a summer and a winter season, seeds have adapted the capacity to enter and exit a state of dormancy. I mentioned a hormone last episode called ABA, or Abscisic Acid, which plays a role in growth regulation by inhibiting cell division and germination. This hormone is largely responsible for stuff like storing nutrients, helping the seed go through its drying out process, and suppressing the seed's germination response. There are also hormones I talked about called gibberellins, which do the opposite. They promote growth, and they encourage germination. For dormancy to be cancelled or shut off so that gibberellins can be expressed and the seed can germinate, it has to undergo some kind of activation or meet some kind of threshold. Some seeds need to be exposed to red light, which indicates that there's a relatively decent amount of sunlight available. Other seeds have really thick coats, and so light can't get through them. And to germinate, their coats need to be scarified, or scraped and degraded and worn down in some way so as to expose the internal tissue to sunlight and fresh air. Some seeds need a temperature cue to end dormancy and initiate germination. like the seeds of boreal plants that need to know when winter ends and when summer begins. Then there are plants that live in areas prone to wildfire, which also includes a lot of boreal plants. These wildfires will clear out the underbrush, removing all of the local competition for water and sunlight, and creating a nutritious layer of plant ash. These conditions are pretty good for plant growth, and so this is an opportune time for the seeds to germinate. And the heat signal from the wildfire itself has evolved to be an appropriate signal for germination. All of these germination conditions require water. Water is the necessary key ingredient. All seeds need it. Germinating when you aren't near water is stupid. It's a death sentence. So water is like the last signal necessary, the final failsafe that's needed to germinate. When water gets into the seed, it rapidly fills the cavity and flushes through the plant tissue. The availability of water in the embryo tissue allows the seed to start consuming oxygen in cellular respiration and producing proteins for growth. After some time, water uptake will slow to a halt. The plant is as hydrated as it needs to be. This is when the cells really come alive. Proteins are synthesized, mitochondria fire up their ATP engines and begin multiplying, and the membrane expands as the cells grow. By now, the cells are flush with water and the embryo is ready to germinate. The root tip, the radicle, emerges first from the seed and reaches around to find soil. The root digs in and grows, penetrating deeper in the soil and anchoring the plant to the ground. The stem rises and lifts the cotyledons up into the air. The new leaves unfurl in the sunlight. Water is absorbed, carbon dioxide is inhaled, photosynthesis begins and creates sugars, and the young plant begins its life. And that about does it for plant reproduction. That's more or less how the whole process goes. Plant reproduction is really involved. It's a really intricate process, tailored by evolution to allow the plants to respond and time their growth in accordance with the growing seasons and other environmental stimuli, so as to maximize their chances for survival and reproduction. Join me next week for the final episode in this series on plant physiology, where I'll explore the fundamental chemical process of photosynthesis, as this is the process that enables not just plant life, but all life on the planet to live and thrive. And as always, thanks for listening.
Would you like to support the BioLogic podcast? It's super easy. When you open a new episode, press the like button or share it with your friends. If you aren't subscribed, you should hit the subscribe button so you can enjoy a new episode every week. You can also peruse our official store, which has a ton of cool stuff like hand-designed t-shirts, hoodies, and stickers. All the links you need are in the description section below.